The Human Genome Project was an international partnership that operated from 1990 to 2003. The foundation of genetics were based on Gregor Mendel. Mendel was an Austrian botanist in the mid-1800s. He studied bees in his garden and concluded that offspring inherit a form of genes named alleles from each parent. Shortly after, a Swiss physician named Friedrich Miescher isolated the nucleic acids which is associated with DNA. 60 years later, a woman named Rosalind Franklin created a photograph of the helix-shaped DNA. This led to the next group of scientists who worked with her. Scientists James Watson and Francis Crick worked with Franklin in 1953 and created a model of the molecular structure of DNA, which traced genomes. About a decade later, an American biochemist named Marshall Nirenberg cracked the genetic code by matching amino acids to synthetic triplet nucleotides. In 1972, another American biochemist named Paul Berg created a recombinant DNA, an artificial DNA that was formed by combining different organisms' constituents, which made gene cloning standard. Right after, in 1972, doctors Walter Gilbert and Fred Sanger created new methods of sequencing a genome, the Sanger method, to determine the order of bases. All 5,386 bases of the bacterial virus were sequenced. About a decade later, Doctor Kary Mullis and his team created a method where small minuscule samples of DNA could be multiplied. This was the invention of polymerase chain reaction technology for amplifying DNA. The next year, Leroy Hood, an American biologist, created the first machine that could sequence more than a million bases in a day. With new technology and methods of sequencing, scientists collaborated and created the Human Genome Project to sequence the entire human genome. The project received interest from the Department of Energy and received funds. By 1988, the National Institute of Health created the Office of the Human Genome and was sponsored by many health and science foundations. In 1996, all the funded scientists met in Bermuda to publicize the sequence data through an agreement named the Bermuda Principles. This this made the sequence unable to be patented. In 1999, scientists finally announced that they had sequenced completely the chromosome 22, the smallest chromosome in a human. Now, more are being done trying to implement the use of genomes into life.
Cloning is defined as a replicate, a fragment of DNA placed in an organism so that there is enough to analyze or use in a protein production. Cloning is enhanced from the genetic engineering and altering of genes. Some scientists see this as an evolution and a big step in science so that they can have a major impact on the lives we live today. Although cloning has its positive impacts, it creates a division between the lines of ethics and science. Anthony McCarthy discussed how cloning destroys the ability to mourn the dead specimen because many more are created. With cloning, each individual being cloned, you'd find that they're valued less because they can be multiplied, thus losing the individual's value. The World Disease Weekly almost mentions the point that cloning of fetuses on humans have not been proven to be safe, causing genetic mutations in DNA. Because some genetic mutations can result in physical and mental anomalies, it is viewed at the same level as abortion.
Technique in genetic engineering is gene therapy or genetic manipulation, allowing for scientists to alter DNA. In genetic engineering, you have the ability to alter genes to obtain the physical appearance and genetic composition you desire. While doing so, most who try to accomplish this are unaware that they are altering other bodily functions. If you were to alter your body over its capacity, you could stress other organs, causing major damage. Not only this, but it brings up society's trend to fit in. Discrimination would be a major factor because everyone would be trying to live within society's standards. You're trying to change the way people were meant to be. Scientists are basically saying that the child they're genetically manipulating isn't good enough, so they have to make them better. With this technique, you lose your individuality. There are instances where altering genes can help prevent other inherited genes, but you have the opportunity to bring more diseases that can be just as severe.